Researchers from the University of Texas at Austin and the National Institutes of Health have made a critical breakthrough toward developing a vaccine for the 2019 novel coronavirus by creating the first 3D atomic scale map of the part of the virus that attaches to and infects human cells. Mapping this part, called the spike protein, is an essential step so researchers around the world can develop vaccines and antiviral drugs to combat the virus. The paper was published in the journal Science. The scientific team is also working on a related viable vaccine candidate stemming from this research. The researchers have spent many years studying other coronaviruses, including SARS-CoV and MERS-CoV. They had already developed methods for locking coronavirus spike proteins into a shape that made them easier to analyze and could effectively turn them into candidates for vaccines. This experience gave them an advantage over other research teams studying the novel virus. Just 2 weeks after receiving the genome sequence of the virus from Chinese researchers, the team had designed and produced samples of the established spike protein. It took about 12 more days to reconstruct the 3D atomic scale map called a molecular structure of the spike protein and submit a manuscript to the journal Science, which expedited its peer review process. The many steps involved in this process would typically take months to accomplish. The molecule the team produced and for which they obtained a structure represents only the extracellular portion of the spike protein, but it is enough to elicit an immune response in people and thus serve as a vaccine.